I'm just going to record the antiviral and antihelminthic drugs for you. We'll talk a little bit about antibiotic resistance and MOAs of that, and then we'll be done with the with chapter 20 on antibiotics. So antiviral drugs are really difficult to come up with. Just think about, I mean, the situation we're in right now. It's been very difficult to come up with treatments against COVID. And this is because viruses themselves really don't have a whole lot of targets. If you think about it, the structure of a virus is really just a protein capsule. Inside of that capsule, that inside that little protein box is just some genetic material. There are a few viruses that do bring their own enzymes with them, like HIV brings 3 different enzymes with it. That makes the HIV target a little easier to find. And then, of course, we have the protein spikes, and that's really about it. So there aren't plasma membranes and cell walls and ribosomes and polymerases or things like that to target. This makes coming up with antiviral medications fairly difficult. Also, because viruses utilize our own cells for manufacturing, they're using a lot of our own cellular components. Many antiviral drugs can also have some severe side effects. So we're just going to take a look at a few. Most of them have actually in the past. Historically, most of them are against HIV. That's when the antiviral medications really kind of made a big boom on the market. But now there are several others for other types of conditions. Most drugs will be harmful to the host. They do have fairly limited uses, and most antiviral drugs are fairly targeted. Most of them will work as an analog to viral DNA or RNA. Most of the time, viral DNA and RNA, the genetic material found inside of viruses, is just has a few structures to it that our own DNA does not. Remember, we have like pattern recognition receptors that recognize viral RNA and DNA. So we are able to kind of target the genetic material itself. There is a lot of research going on for other targets, but right now the best targets are the actual genetic material. Now, in the antiviral drugs, what we mean by targeting the nucleotides is these are called nucleoside and nucleotide analogs. An analog is a chemical compound that is going to resemble a natural compound. And in this case, we're talking about the drug acyclovir. Acyclovir was originally created against HIV, but acyclovir is actually used for herpes infections. So it's used against the herpes virus. Acyclovir mimics deoxyguanosine. Deoxyguanosine is a nucleoside. A nucleoside is before a DNA molecule has been incorporated into an actual DNA. So here we have what's called deoxyguanosine. This is the G in ATCG of DNA. And what this drug does is it mimics the guanosine, and you can see that it has the same guanine, the same purine structure as guanine. It looks exactly like it. So DNA polymerase is going to recognize that guanine, the nitrogenous base, and try to incorporate it into, during DNA replication, try to incorporate it into the DNA or into the RNA. But in this case, because this is DNA, because it's deoxy, in this case, the actual deoxyribose base, that sugar phosphate backbone, cannot be formed because acyclovir doesn't have a deoxyribose sugar. It has a completely different base. And so the base pairing will occur between a G and a C, but then the sugar phosphate backbone that follows cannot be made. This will disrupt a DNA replication. It'll happen multiple times. DNA polymerase will stall, and the viral DNA cannot be replicated. Antiretrovirals have a couple of extra targets. Now, like remember I said HIV, for example, has the ability to bring some of its own enzymes with it. And in this case, there are other nucleoside or nucleotide analogs, and antiretrovirals are often targeted against HIV in particular. Now, here's acyclovir again, and you can see in this particular diagram, normally our guanine nucleotide would be incorporated directly into the DNA. DNA polymerase would recognize it. It would be able to use it for base pairing. It would work. Here, this is called a thymidine kinase. It is in a virus-infected cell. It's going to take that sugar phosphate backbone, and instead of taking guanine, it's going to use acyclovir. Because you can see that structurally this does not bond properly, DNA polymerase gets blocked, and it's going to fall off, and DNA replication will stop. Now, in antiretrovirals, again, these are produced mostly against the HIV, the human immunodeficiency virus, and two of those are tenofovir and AZT. AZT was actually the very first one. This is because HIV brings with it a couple of enzymes, and one of them is a reverse transcriptase. Reverse transcriptase, if you remember, retroviruses, instead of going from DNA to RNA to protein, they actually introduce RNA that is reverse transcribed into DNA, which is then transcribed into messenger RNA, and then eventually translated into proteins. Because of this retro process, this RNA to DNA process, these guys have to bring their own enzyme. And so the enzyme they bring along with them that does this is the reverse transcriptase enzyme. So if we can target that reverse transcriptase enzyme, then the virus will not be able to reverse transcribe its RNA into DNA. Enzymes are targeted, again, the most common HIV targeted enzymes are going to be reverse transcriptase, which was on the previous slide, but I want to add it here. So reverse transcriptase gets targeted. The enzyme that's responsible for integrating the viral DNA that's been made by the reverse transcriptase, the one responsible for integrating that is called integrase. And HIV integrase can also be disrupted. So here's the integrase. And then protease. HIV, in order to get everything started once it infects a cell, actually brings its own enzyme with it to kind of kickstart things. It's called a protease. And what the protease does is it breaks down host proteins and helps to activate viral proteins, as well as break down the capsid around the HIV genetic material in order to get the HIV genetic material where it needs to be to get initiated. There are also a few drugs out there that are not necessarily targeting the proteins or enzymes that are brought with HIV, but are actually inhibiting fusion. Remember, many viruses enter cells through fusing their envelope with the plasma membrane of the host cell. Well, if we can prevent viral fusion, then the virus can't even gain entry into the cell. If it can't gain entry into a cell, it can't replicate. Other antiviral drugs that are enzyme inhibitors are the protease inhibitors, so they're usually used in combination with other retroviral drugs, inhibiting attachment as well as fusion, inhibit uncoating. Maybe we can stop that uncoating process so the genetic material can't get outside of the capsid. And then also increasing interferon, either interferon efficacy or efficiency, or increasing interferon production, promoting that interferon production so that our own immune system will, one, respond greater, and two, will inhibit fusion and entry into neighboring cells. That brings us to the antiprotozoans. Now, the antiprotozoans are probably the most difficult to come up with because protozoans are single-celled eukaryotic organisms. They are very similar to animal cells, so they have a lot of targets and oftentimes have many side effects. The first is chloroquine. Chloroquine was the original drug used for treatment against malaria, but now we're starting to see a lot of Plasmodium resistance to chloroquine. It inhibits the synthesis of DNA so that these organisms are unable to replicate and go through asexual reproductive processes. These guys do go through cell division, although it's not binary fission. It's not usually I forgot the word. Identical. One example of this is mefloquine. Mefloquine is used when chloroquine resistance is found in some Plasmodium. Mefloquine can be used, but it does have some really potent psychiatric side effects. So people can get can go into some psychosis things like hallucinations, mood changes, behavioral changes, those sorts of things, even suicidal. So this is one of those cases where sometimes where the treatment is worse than the actual disease. Dioda is another antiprotozoan that is used against entamoeba infections and other types of amoebic infections. One problem, however, is it does, for some reason, interrupt function of the optic nerve and can lead to either permanent or temporary and partial blindness. Then we have flagell. Flagell is actually very common, and in addition to being an antiprotozoan, it also has some efficacy against bacteria. Flagell is oftentimes given in combination with other antimicrobial drugs. Flagell is also given in not only in parasitic infections, but also in C. diff infections, many times because flagell may be used as a preventative. Somebody with C. diff is going to have very little gut flora, if any, so they are very open to infection by not just by bacteria, but also by other protozoans. So flagell does have some efficacy against C. diff. It also can be effective as a preventative for other protozoan infections. And flagell is a zoonotic drug. It's given to other animals besides humans. Niclosamide prevents the generation of ATP. It's used specifically for tapeworm prevention. And then praziquantel is used in the flatworm and flukes. Praziquantel will make flatworms and flukes very fragile, so that when they get attacked by the immune system, they're much easier to be destroyed. They can also affect the autonomic nerves. That maybe many worms actually have a primitive nervous system, and so causing paralysis will prevent the worm from being able to attach, and it just kind of gets stuck in the intestines and then eventually will get passed in feces. Mebendazole is a broad spectrum paste that's used quite a bit for intestinal roundworms. It prevents them from being able to absorb nutrients, so it basically starves them out. Mebendazole is sometimes found. It's called parental pomoate. It's the strongid paste. We used to use this, and it's used in horses quite a bit. It prevents the formation of microtubules and inhibits nutrient absorption, so that any worms would get passed out, usually passed through feces. It's also given for it's a deworming paste. It's the yellow one that your dog gets. It smells like bananas, and it's used for dogs, cats, horses. It's used in the agricultural industry quite a bit. Then there's ivermectin. Ivermectin is a nerve agent. It does cause paralysis of any round. It's usually used for intestinal roundworms. It's extensive. It's much stronger than the strongid, and it's used quite a bit in the agricultural and pet industry. Used to use ivermectin in horses quite a bit too. It's more often used in horses and cattle. One problem with ivermectin, however, is you cannot give it to cats. Ivermectin cannot be given to cats. It affects the optic nerve and can cause temporary blindness. It can cause permanent blindness if you continue to give it to them. So do not give ivermectin. Both ivermectin and strongid in some pet stores are actually sold over the counter, particularly strongid. I'm not sure if you can get ivermectin over the counter anymore, but strongid you used to be able to get in some pet stores that sold things for horses, for large animals. And people would buy these wormers for their dogs or cats. It's the same ones. They would just use a much smaller dose. And ivermectin, however, can cause blindness. It's toxic to cats, so you never want to give it to any kind of cat or feline. Now, antibiotic resistance is something that's a really big problem. We're seeing major increases in antibiotic resistance throughout the world, and we're seeing bacteria develop resistance at a much faster rate than we are seeing new antibiotics being produced. This has become a really big problem, and it's such a big problem that here in the United States, the NIH provides millions of dollars in grants every year to multi-billion-dollar pharmaceutical companies to get them to research and create new antibiotics. Antibiotics tend to not be big money makers, so most pharmaceutical companies don't spend a whole lot of time coming up. They don't want to invest a whole lot of money and time into a product that's not going to bring them big profit margins like some of the other drugs they come up with. So the NIH says, well, hey, we'll pay for the research if you will do the research and come up with a new antibiotic. And so some companies have taken them up on that and some have not. Some have said, hey, it's just not worth it. We're going to keep our resources somewhere else. There are a lot of different types of mutations that can lead to antibiotic resistance, but why would an organism go through all of that evolution in that process unless they were constantly being exposed? So some of the mechanisms of antibiotic resistance that are developed are due to a couple of different factors. One way that microorganisms will actually develop resistance is they can evolve a enzyme, create an enzyme, or take an existing enzyme function and add to it the ability to destroy a specific drug. Beta-lactamases are an example of enzymatic destruction of the drug. It just breaks that beta-lactam ring and destroys it, and then the penicillin is no longer effective. Some organisms will prevent penetration of the drug. So just the lipid layer alone on many gram negatives makes them impenetrable to some drugs. This would be an intrinsic form of resistance. Some will alter the drug's target. So they say, okay, well, if you're going to send this drug in and it's going to target this particular enzyme, if they change the shape of the enzyme, it's still functional for them, but the drug can no longer attach to it, then they alter the target site. Another form that's really very common is rapid ejection of the drug. This is done through a structure called an efflux pump instead of an influx pump. And what happens is the drug gets pumped out of the bacterial cell as fast as it got pumped in. Pseudomonas and E. coli are notorious for evolving efflux pumps to pump drugs out as fast as they try to penetrate into the cell. The genes for this type of resistance are usually found on plasmids, and plasmids, if you remember, are a form of genetic diversity. So bacteria are able to share these recipes of resistance with each other very rapidly. And because of that, we can get all new strains of bacteria that become very resistant very, very quickly. Most multi-strain resistance factors are usually found within hospitals. They'll start out in hospitals. MRSA started in hospitals. VRSA started in hospitals. MERS, the CREs, the carbapenem-resistant enterics, which are a big problem now. All of these different strains of resistance have really begun in hospitals, and that's not because hospitals are necessarily doing anything wrong, but that's where all of these microorganisms are concentrated together in the same environment. So they have that ability, that opportunity. There's greater opportunity there for those microorganisms to mix and match and start swapping information like a big recipe swap. What causes them to develop this resistance? Why are they coming up with resistance to compounds that may not even exist yet? Well, part of it is because we don't use them correctly. So the misuse of antibiotics is going to not just introduce resistance, but what it really does is it selects for organisms that are already resistant, so that the population of that microorganism, now it's a greater population of mutants than it is of the wild type. The wild type are still susceptible to the antibiotic. So we use the antibiotic, we get rid of the wild type, and what's left are the mutants that have become resistant to that particular drug. What are some types of misuses that actually aid or help develop resistance, or even, I should say, promote resistance? Well, one is using outdated or weakened antibiotics. Antibiotics, like any other drug, have a shelf life. So if we use an antibiotic that is really old, it's been sitting around, it has been weakened, it has broken down over time. So in this case, we might think that we're giving a particular dose, but we're really not. We're just giving a really weak, low dose that won't necessarily kill the microorganism, but it does introduce it to that actual antibiotic. And any organism that has just a slight mutation that makes it just, a weak resistance will survive. Those that have zero resistance will die. What happens then is we're left with a population of bacteria with weak resistance, and they eventually can strengthen that resistance over time. This is why the strength or concentration of the antibiotic needs to be appropriate to the microorganism that is being treated. Using antibiotics for viruses or protozoan infections when it's not warranted, using an antibiotic for something inappropriate. Now we're just introducing normal flora to antibiotics for absolutely no reason, because the antibiotic is not going to be effective against a virus. You don't use antibiotics against viruses. Using antibiotics in animal feed. This was found to really promote the resistance of things like gram-negative bacteria against penicillins. Antibiotics for years and years have been put in animal feed to prevent our animals like cattle, pigs, chickens, to prevent them from developing infections. In this case, the antibiotics or antimicrobials were being used as a preventative as opposed to a treatment. This would allow farmers to grow more animals, to raise more animals in a smaller area. There are restrictions to how many cattle per acre a farmer can have, or how many chickens per cage, or however they do chickens, how many they can have per area. And by introducing animal feed with antibiotics in it, these farmers were able to kind of cram more animals in a smaller space. Otherwise, disease would wipe those animals out. But because they're all on antibiotics, they don't get those diseases. The problem with this is the beef that we eat, the pork that we eat, the chicken that we eat now has antibiotic metabolites in it. Though the antibiotic itself may not be present, but metabolites of that antibiotic will be introduced to our own normal flora, and then we'll start seeing resistance can start developing to some of those metabolites. Failure to complete a prescribed regimen. This is pretty straightforward. If I kill off all the really weak ones and then, which might be the majority of the population, I start feeling better, I stop taking the antibiotic. What's left behind are those weakened ones. The same idea, this is the same idea as using outdated or weakened antibiotics. So now the resistant ones are left behind because I didn't expose them at a high enough or long enough concentration. And so now those organisms can grow back, and now I have a population of resistant microorganisms. And then finally, using somebody else's leftover prescription. Like I have stated in class, you would not do this with any other medication. So why would you do this with an antibiotic? You may not have the same strain or even the same class of bacterial infection that someone else has. So their penicillin, if you have a gram-negative infection and you take somebody else's ampicillin, it's not going to do anything for you except introduce ampicillin to all your flora. And so now you're just spreading the ability, or you're spreading the exposure to antibiotic to organisms to develop resistance. Now, one of the things that's being looked into is how can we use other forms of antimicrobial agents? Does everything have to be an antibiotic? Can we change the target by changing the drug? So some of the ideas are antimicrobial peptides. These are broad-spectrum antibiotics that, instead of being derived from soil organisms or fungi, are actually being pulled from plants and animals. Animals in particular do have some ways, some of their own ways of preventing bacterial infections. Sharks never get sick, and so they actually have a squalamine protein produced by their immune system that prevents translation. Protegrin and manganine are two proteins found in pigs and frogs that also have antimicrobial activity and are being researched for another source of antimicrobial compound. Another is the antisense agents. Antisense agents are a complementary DNA or RNA that can be given that will bind to the RNA of a virulence gene so that that RNA cannot be translated. So this is going to, in an antisense, right here, can prevent translation. If it's an antigene, they call it a decoy, then it'll inhibit transcription. And then whatever protein is being targeted will not be made, and the bacteria wouldn't be able to survive. This antisense agents have a lot of promise because they can be really, really specific to gene sequences. And that can be very helpful when it comes to not just bacteria, but also against viruses, protozoan infections, and fungal infections. The last is phage therapy. Phage therapy would be bacteria specific. We're talking about introducing phages, bacteriophages into the body in order to target bacteria specifically. There's still a lot of research, although it's been very effective in some applications. One of the problems, however, with phage therapy is that our own immune system will recognize it and destroy it. So dosages and how to get it delivered where it needs to go is definitely part of the research that's going on. But there have been some clinical trials with phage therapy, particularly in like diabetic neuropathy, which are like wounds in the feet and that are infected, severe infections. There has been some really positive effects of phage therapy. So that's it. That's the last of the antibiotic chapter of chapter 20, and I'm looking forward to getting into some epidemiology with you. Have a good one.